In another video, I'll review the various structures of the human endocrine system, the hormones that they make, and the effects that they have. This is going to be a much shorter just treatment of just the evolution of some of these structures. And it's going to be shorter because the foundation of the endocrine system is that there are molecules released hormones, which are signals. And so if one were to then compare, vertebrates, humans, et cetera, and ask, where are the homologies, where are the similarities, it becomes very molecular very quickly. And so, for example, you would say, oh, well, estrogen, testosterone, progesterone, aldosterone, these are all a group of hormones known as steroid hormones. So which animals make these steroid hormones, which animals make receptors for these hormones? And then, you could comment on, oh, well, this is in the same group as the molting hormone in insects, ecdysone, and one could talk about gene duplications, which then lead to an amplification of the number of receptors, because that's a big thing. You could say at this point there seems to be one steroid hormone being used as a hormone and one type of receptor, but then there's a gene duplication, and now there's two different receptors, and another gene duplication, now there's more receptors, and so therefore what we now refer to as the steroid hormones, it belongs to a gene family which has been amplified. And so once again, that's very molecular. And in this treatment, which is going along with a comparative vertebrate anatomy course, just a little too molecular for the main treatment of the course, which is more macroscopic anatomy. Certainly lots of tissues make hormones. So adipose cells can release hormones. White blood cells can release, hormones. But once again, now we're more in the, the cellular nature of this. If you look here at this oocyte, here you can have different layers of cells releasing, hormones which are acting very locally. And so the endocrine system really lends itself to a treatment of more like a cellular and a molecular study rather than, an anatomical study. And so just some quick comments on the history of our endocrine system. The first is most receptors are not the receptors I just mentioned for the steroid hormones, which then bind to DNA and start transcription. So that's a family of genes. So there's a family of genes which after duplications now could perceive estrogen and testosterone, progesterone and aldosterone. Most receptors are what are called G protein coupled receptors. For most of the hormones of the human body, both the long distance acting circulating hormones, and then also a lot of the more local hormones. Well, this goes back to the very first living things. Bacteria were the ones who first developed G protein coupled receptors. And when you look at how these receptors are acting, say in yeast, in primitive eukaryotes, the reaction with the mating factor in this one-celled organism is kind of reminiscent of the signaling using hormones in complex animals. And so, some of this molecular aspects goes, back to early living things. And then over long periods of time, there were gene duplications, which then amplified the number of G protein coupled receptors, the number of those receptors for the hormones. And so thus, later organisms had a much greater repertoire of hormones and receptors than would have been present ancestrally. And so thus we can then trace those. And you see here are all members of the same gene family. In humans, G protein coupled receptors, this is actually the largest family of genes in the human genome. So we could certainly study the evolution of the endocrine system with that in mind. We could also then focus a bit more on structures as well. So just let me zip through some of these. The nuclear hormone receptors, they are known from invertebrates, and they then started to, to duplify. There are invertebrates which make insulin, but then here you can see that, insulin making undergoes stages. So insulin or insulin-like molecules, they date back to invertebrates. Later ones then are not only making insulin, but they're making it from cells along the gastrointestinal tract. And then there are clusters of cells known as islets, which then make insulin, and then still later this is incorporated into the pancreas. And so a pancreas, which has endocrine cells making insulin, isn't how insulin first appears. First, it's made by cells in general, then cells in the GI tract, and then by these islet cells, and then these islets are associated with the pancreas, and that last step doesn't occur until the jawed vertebrates. In the same way, there are adrenal glands in the jawed vertebrates that are near the kidneys, but at this early stage, the tissue that we find in the medulla of the adrenal glands, which release epinephrine, is not yet incorporated in it. And so there are a couple of glands which are more composite structures in humans, where they have this tissue that maybe makes this first hormone, another tissue that makes another hormone. Well, they could have started off as separate tissues in different parts of the body in ancestral vertebrates. And only later in evolution do they end up being associated with each other. And so clearly we can see, changes over time, but once again, it's more on the molecular level, more on the cellular level, or, these tissues are now associated with each other, and not quite what is, typically studied in, a comparative anatomy class. So one more example of this. So oxytocin is incredibly important for mammals. All right, so it's the signal for orgasm, obviously, associated with the higher brains, like the septum region of the cerebrum. It causes uterine contractions during childbirth, which we associate with mammals giving live birth. It causes milk letdown, which once again we associate with mammals. But the interesting thing is that this is actually belonging to a gene family, which goes back to invertebrates. Everything that I just said is associated with reproduction in mammals. But hormones of this gene family, which have the same number of amino acids, certain amino acids, the same ones at specific spots, which affect the folding, that are involved in reproduction, in a number of invertebrates. And so what we see very often is, even though we associate this hormone with some of the functions of higher order vertebrates like mammals, nevertheless, this was a modification of a simpler ancestral system. So something doesn't have to be, like an either or complex things appearing from nowhere. You could have, certain molecules that then, become involved in more and more complex pathways over time. And you can see how homologous these are if you were to study, once again, how these are made, the genetic sequences. So you can compare genetic sequences and find, that these genes form these gene families based on similarities, suggesting that these molecules share a common ancestry, but that they have, evolved over time. Once again, I'm going to move on, but much of these ideas are perhaps better studied in genetics rather than comparative anatomy. The human hypothalamus is amazingly complex, and it is not only a critical part of the nervous system. It is where oxytocin is made, for example, but also a part of the endocrine system, as I just pointed out. And so this is an important part of the endocrine system. And certainly as we compare the hypothalamus of humans to those of other organisms, we can see similarities. And we can also see that the complexity of the hypothalamus has increased over time. And so the more complex functions we associate with mammals, has changed over time. A hypothalamic region goes back even before there were vertebrates. In the primitive chordates, there are areas which are homologous to the hypothalamus. And then as we go up the family tree into primitive vertebrates like the lamprey alive today, the primitive jawed vertebrates like sharks, etc., we can see more and more similarities between the hypothalamus that they possess and those of humans. So the hypothalamus is a major area in the human endocrine system. And here you can see, some of its anatomy, some of its functional roles, certainly go back to earlier vertebrates. So the hypothalamus has evolved over time. The pituitary gland has evolved over time. The human pituitary has two halves, an anterior half and a posterior half, and an important portal system which connects the two. Originally, these two masses were separate. And so when the pituitary begins in vertebrates, the anterior part is developing in the roof of the mouth from an area known in human embryos as Rathke's pouch. So in human embryos, the anterior pituitary is still separate from the posterior pituitary. The posterior pituitary develops as an outgrowth from the brain, and they associate. But as you were to go through different organisms, once again, the primitive chordates seem to have a homologue of the pituitary and some of the hormones which we use. And just wanted to point out, prolactin, once again, we tend to associate with mammals. This is what causes the mammary glands to develop milk, which obviously we associate with mammals. That's where mammals get their name. But prolactin has lots of functions. Fish can use prolactin, and prolactin, or at least a prolactin-like molecule. I'm sorry, I misread that. Prolactin go back even to primitive chordates. And so the pituitary is homologous over the vertebrates and has changed over time. The portal system, where they're in the vicinity of the pituitary gland, goes back to primitive vertebrates, and now by the jawed vertebrates, it connects the hypothalamus to the pituitary gland. So now the hypothalamus can put hormones into the bloodstream, which goes straight to the pituitary. Jawed vertebrates share a POMC gene, which is interesting because it makes a peptide that then gets cut into lots of pieces. All right, and then so each of these then is a separate hormone. So this is not just something we see in humans. We see it in sharks. We see it in bony fish, etc. By the osteichthyons, the anterior and posterior pituitaries are finally contacting each other. So once again, you can see that there are changes in the pituitary gland over time, and we can say this is a trait shared among the jawed vertebrates or among the sarcopterygians, the fish, and the amphibians, etc. That's not just true of the pituitary gland and the hypothalamus, but also of the thyroid gland. Now, we can associate the thyroid gland with some of mammalness because it's the hormone thyroxine which causes us to shiver and helps us to be warm-blooded and maintain a constant body temperature. But a homologue of the thyroid gland called the endostyle goes back to the chordates before there were even vertebrates. Another thing that we see here is, in humans, the parathyroid glands are nestled within the thyroid gland, and the thyroid gland contains both the cells which are releasing thyroxine and the cells which are making calcitonin. In vertebrate ancestors, these were three separate masses of cells, which then gradually accumulate. And so once again, as we discuss homology, you can see that, you can have cells making hormones, but, their associations into a larger glandular structure can develop over time. Even going back to these hemichordates, the association of iodine with the endostyle to be bound with the thyroid hormones then predates then the vertebrates. And so the thyroid gland is an ancient structure. And then as we go forward, we can see that the various separate tissues get associated. Some of this happens after the gills are lost as vertebrates, go to adapt to land, because once tetrapods no longer need gills, they reorganize this area. And it's, some of the reorganization of this area that ends up putting the cells which are making calcitonin and the cells which are making thyroxine together. And so the thyroid gland started off as different masses of cells. And in jawed vertebrates, those two masses of cells making calcitonin and the thyroid hormone thyroxine, they end up in the same place. And once we get to amphibians, then we have the parathyroid glands associated with them as well. And so the glands which mammals use to regulate their blood calcium, but also which they use to help keep warm in an endothermic metabolism, this then is a modification of the roles that they had in simpler organisms. As I've described elsewhere, when we think of the pineal gland, we think of it as a gland in the endocrine system because it makes melatonin. It takes tryptophan and then converts it into melatonin, which is released at night. But the pineal gland has a fascinating history because in the earliest vertebrates, there was a hole at the top of the head with translucent skin covering it so that light would hit the brain where the pineal gland was. And so that the pineal gland's ability to make melatonin at night was tied to the fact that it was actually measuring the amount of light in the as light came into the skull. And so the pineal gland was sometimes people refer to as a third eye. Now, it never would have participated in vision. All right, so this was a circadian function, just detecting light to determine a day-night cycle. But this gland, which makes melatonin on a day-night cycle, was once part of a photoreceptor organ, which actually detected light. While mammalian pinealocytes no longer detect light because they're no longer a hole at the top of the head, and our big cerebrum has grown over the diencephalon. Our cerebrum is, so expanded in mammals. But the pineal glands used to perceive light, and our pineal glands release melatonin, but that's true of the pinealocytes which perceive light. And even things like rods in the eye make melatonin. It's just they don't make it as a hormone which travels. They make it as a locally acting signal. And so the pineal gland, was modified in the ancestors of mammals, which finally closed this pineal foramen, and they developed a cerebrum which now grew so large that it covered the pineal gland. So just to wrap up, the endocrine system doesn't lend itself as nicely to a comparative anatomy study as many other structures. I mean, the pituitary gland is the size of a pea, for example. The pineal gland, while it's here, once again, not a macroscopic anatomical structure. But nevertheless, if one were to study it, one would see its evolution, not only in the genes for the hormones, and the receptors and the amino acid sequences of the proteins, but also the cells which are releasing these hormones, their association into tissues, which tissues associate to make endocrine glands, the various functions of these, with mammals adding additional functions to hormones like thyroxine or prolactin or oxytocin, which didn't originally exist. And so the human endocrine system, which I describe elsewhere, has its roots in more primitive vertebrates.